Thank you, everyone, for allowing me to explain you my work. I'm Silvia Locubiche, and I'm a predoctoral student with Xavi Altafag. And I'm going to show you a little bit of my doctoral thesis project, but mainly I want to drive you through my results or what we are trying to do with the zebrafish by knowing the model of the zebrafish. So to start, this is our main character today. This is an adult zebrafish. Daniel Rerio is the scientific name. They are 2-3 centimeters long when they are adults, and they are this long or this adult form within 2 months, where they start laying eggs, and we can start having more animals, and they have a life expectancy of about 3 years, so we can really use them for a long time. And you may be wondering why zebrafish. It seems quite weird. It's not a mice or a rat that we are more used to it, but I will try to convince you that they are a really good model for research. They have several biological characteristics that are quite interesting. First of all, they are fully developed, or they have all their systems developed and functional at 5 days post-fertilization. So we don't need an adult or a big mice to work with them. We can already work with them after 5 days. And interestingly, and it's quite surprising, their genetics are super similar to humans. We know they are not mammals, they are not mice. It's quite complicated to think about it, but they are still vertebrates, and they have up to an 82% homology of human genes related with disease that we can study in the zebrafish. And also their genome is really well known, so we can use different genetic tools that are really well known nowadays to work with them and to understand several genetic diseases. Also, their physiology, I know it seems weird, but it's quite similar to humans. For example, the heart rate and how the heart works is more similar in zebrafish when compared with humans than with mice. So they are really good model for that also. And some organs are super similar also to humans. It doesn't seem like this because they are quite different, I know, but as you can see in the colors, the main structures are conserved, and what we see in the zebrafish can be moved carefully to humans. This is how they look. These are the adults, and these are the larvae, 5 days post-fertilization larvae that we work with in the lab. And they have several advantages when compared with other animal models. They are easy to manipulate as they are so small. We can just pipette them into different plates. And this is the size of a normal plate. In these plates, we can have up to 96 animals. So we can perform a lot of experiments with a lot of animals in short times. They have a fast vitary cycle. As I mentioned, at 5 days, they are already functional. And in 2 months, we have adult animals that are laying eggs, and we can start our experiments. The fertilization is ex utero. This means that it's the fertilization and all the development, it's not inside the mother, so we don't need to kill any mice mother. Or we can study everything just directly outside the animals in the petri dishes and in the incubators in the lab, so it makes our work way easy. We have a large amount of individuals, meaning that for each cross of one male and one female fish, we can have 100, 200, 300 eggs, so we can perform a lot of experiments. And what I think it's super important in this kind of research that we are working with rare diseases, is that they are super economic and cheap, both in money, because we are wasting less space to keep them, we are wasting less amount of compounds, but also of time, because we're working with them when they are super young, and also we are treating them with different therapies directly in the water. So we don't need to inject anything to any animal. It's super fast to do, and it's not really that time consuming. Here you have the development of a zebrafish egg. It's quite interesting. I love these images. This is the one single cell stage. Then we have the yolk, and you will see that this single cell will start dividing. Then we have 2 cells, 4 cells, 8 cells, and go on. And the yolk is where they have all the nutrients that the cells need to keep dividing and to keep growing. And this yolk allows us to have these zebrafish living without the need of eating up to 10 days. So we can have them in the lab without the need of feeding them to perform our experiments. And well, you see it starts moving and then all the parts starts forming. This is like 16 hours post-fertilization. We can see the eye in here and everything is getting formed. You can see that the yolk is getting smaller because everything is being consumed. And they are inside this chorion, this capsule. In here they have between 1 and 2 days, they start moving. And in here they are between 2 and 3 days. They have pigmentation, they are inside this capsule, and they have enough strength to break it and start swimming freely in the plate. That is what is going to happen now. They are little aliens. I know it seems weird, but it's so nice to work with them. And as I mentioned, they are really good models because I know it seems weird, and I keep saying this because it seems weird to me after 4 years working with them, but still. With them, we can do several things that we can also do with other models. They have really well-conserved targets and pharmacology when compared with other mammals and with humans. It's not a perfect model. We know that they are not mammals, but it's really a good start. They also have conserved the metabolism of these drugs, of these therapies, when compared with other mammals. And as I mentioned before, their physiology when compared, for example, with mice, that it's what we always compare the zebrafish with, it's quite similar, more similar to humans than this other model. Another advantage that I also mentioned is that it's more economic to work with them because with just one individual, we can do a lot of things, and we don't need different groups of animals to perform each one of these parts because they allow us to do it at the same time. They have organ-specific toxicity, and now I will show you the image of the zebrafish. We can see all the organs easily with the help of a microscope. We can assess behavioral toxicity. A lot of behavioral tests are already set up and are really, it's really interesting to see how they are moving, quite similar to mice, or they have some things that can be similar to what we see in patients. We can do phenotyping of the different systems. We can do perform metabolomics analysis and toxicity analysis. And also, since they are really well known genetically, we can modify them genetically. It's a bad video, I know. I filmed it with my mobile phone in the lab, but it's just for you to see how we do it. We just cross one male and one female fish. They fertilize the eggs, and with this egg, with this one cell that you already saw, but it's this big cell, what we do is to inject with a needle with all the technology to genetically modify this fish. We inject this mix of genetic tools, and we let the fish grow to have our mutant models or models of disease. But it's not only for models of disease. We can also inject other things, for example, fluorescent proteins, as we do with cells, or as we have seen these days in some images. As I told you, and these are the zebrafish. This is a 5-day zebrafish. They are transparent through development, which allow us to see all these systems, not with the bare eye, but with a microscope, because they are quite small, but we don't need anything else. And we can see that they are fully formed. They have the eye, they have the central nervous system, the auditive system, the gut, the heart, the liver, the cardiovascular system. We can see everything, how it's working. And combining this transparency with this ability to genetically modify them, we can find some things just as expressive as this one. This is a model that we have in the lab that has 3 different fluorescents: the heart that is beating in green, the blood cells that are circulating in blue, and the blood vessels that are in red. And this allows us not only to see this, that it's really wonderful, but if there's any problem in these systems or in the heartbeat or something like that, we can easily study them with the animal alive. That it's quite complicated to do it with other models. And you may be thinking that, yeah, GRIN patients maybe don't have these cardiac problems. Well, my models these are one of my models. They are genetically modified, so they have the CNS in red. All the cells, the nervous cells are in red. So if we have any big issue with these cells, will be easily seen under a fluorescence microscope. And having introduced a little bit the model, that I know it's weird, but believe me, it's really nice. We want to use it, and we thought that what we really want to do with GRIN-related disorders is to perform this precision medicine. Everything starts with the genetic diagnosis and the family's contact. In our case in Barcelona, it's Natalia and Angels. And our final end is to find a treatment for these patients. So after the genetic diagnosis, or so far it's been like this, Barcelona's Mireia, we perform structure and bioinformatics to see what is happening in these mutations, where the mutation is, what if it's affecting the channel or the ligand binding domain or whatever. Afterwards, it comes Xavi and his group with the in vitro studies, molecular and cell biology, and biophysics, also with mice models. And the idea of these studies, and we all know that, is to stratify these GRIN mutations mainly in gain and loss of function. We know it's not that easy, that we have intermediates, but the main idea is that if we have a gain of function to attenuate this function, if we have a loss of function to enhance these receptors. And in the end, with the help of this MetaGRIN database and with the predictive algorithm that we've been talking these 2 days, once we have a GRIN variant without having to do all these experiments every time, to finally have a functional assessment and, if it's possible, a therapeutic strategy. So what really concerns us is, as we know, the therapy. And this is where the animal models, mice, enter, but also the zebrafish, and in our case, mainly to do drug screenings. And this project is being performed in ZClinics, that is a company that allowed me to do the PhD with them, and I'm really happy with them. And we're a big group that we all work with Zebrafish, so we can collaborate between us, but this project is performed mainly by me and with the help of Xavi, and Javier Tarrientera, it's my director. The main idea of this project, or the overview of this project, PhD project, is that we start with the patients, with GRIN-related disorder patients, and knowing the information of which are the affected genes through CRISPR, that is a new technology to modify genetically. We want the genes, we generate the different zebrafish genetic disease models. We generate them in the larvae, but we allow them to grow up, and we have the stable lines as an adult that will always lay eggs. The next step is the phenotypic validation, meaning that we need to see which one of the phenotypes or the symptoms that we are seeing in our patients are in our genetic models to verify that they are a good model to study this disease. And we are doing them mainly in the larvae, 5 days post-fertilization larvae, because we're working with a neurodevelopmental disease and because it's easier to work with them. And once we have some phenotypes, we'll move on to juveniles, but starting with the larvae. And the main idea, as I mentioned, is to go to a drug screening. The advantages I mentioned of the zebrafish allow us to do a high throughput screening, meaning that we can test easily and in an economic way a lot of drugs, that the positive ones, the ones that have a result, will be moved to other animal models, but we can start with so many molecules that it will be easier this way. And the idea is to combine all the information that we have from the phenotypic validation to identify novel targets, to identify new molecules, new pathways that we can modulate in order to rectify, to solve the problems that we've been observing. And the final idea is, of course, to bring this to patients again. And this huge overview, it becomes my 3 main objectives of the thesis that we named like this. It's quite fancy, but it's just what I said. The ZebraGRIN to generate the models, the PhenoGRIN to phenotype, to characterize these models, and finally the PharmaGRIN that would be to find new therapies. As we know, and as we've been talking, the main affected subunits in GRIN patients, or where we find the pathogenic mutations, are the subunits GLUN1, GLUN2A, and GLUN2B. And most of them are loss of function. So for us, it makes sense to start with these 3 subunits and start with loss of function, as there are more patients that we can work with. And something I haven't explained yet, and it's quite interesting, is that the zebrafish, one ancestor of the zebrafish several million years ago, they had as we humans do, one only gene, but at some point, a whole genome duplication happened. And instead of having one green gene, we have 2 green genes for each human green gene. These duplicates our work, but it's also kind of interesting because it's not really well defined how these genes work. So in this project, we are focused in this loss of function. That is what we name a knockout, where we directly we stop the activity of these different genes. And with GRIN1, GRIN2A, and GRIN2B, we now have 2 copies of each of one: GRIN1A, GRIN1B, GRIN2AA to AB, and 2BA to BB. It's quite, I know it's quite tricky, but it's I didn't put the name, so. Okay. The idea is to do these knockouts, these animal models that don't have the activity of the GRIN1A or GRIN whatever A and GRIN whatever B in a single way, so we can analyze what they are doing, because it's not really well known. And by crossing these fishes, we will have finally the double mutant that will be our disease model, because it will be what was happening in the patients. Now, once the models have been generated, it's the time to characterize them. And for the behavior, we set it up as, it's quite simple protocol where we breed the fish, male and female, we collect the eggs, we see which one of them are fertilized, are okay. At 5 days, we count the larvae, we plate them in these little plates that I showed you. We treat them if we need to with the different compounds. And after 3 hours, where they are acclimated, they are okay in the plate, it seems that nothing bad's happening, we record their behavior after a few changes we do in the. We do this in this machine that it's not that big. Honestly, it's like this big. It's quite comfortable to have it in the lab because it's not having a lot of space, where we put our plate with our fish inside, and we have a camera on the top that is recording everything that is happening, and it's tracking every fish individually, and we can measure everything that the fish are doing. And this is how an experiment will be seen when they are in this recording chamber. You see these little aliens are moving, and they are just being tracked, and we can see all the distance they are moved, if they are moving more in the periphery instead of in the center of the well, so we can analyze anxiety, we can analyze velocity, acceleration, if they have an altered movement, and a lot of stuff. And one of the things that I think is very important in this case for GRIN-related disorders to evaluate are epilepsies. In here you have the controls on the top and the epilepsy model on the bottom. When they react like this, that it's what we named the convulsion pattern that they have. It's because we flash them with a light, just 1 second flash, and they react to this flash by having these convulsions. We can track, as I said, this movement, and we can see that they really are having an aberrant movement that can be quantified, that can be analyzed, but also not all the epilepsies look the same as it happens in humans, and different genetic epilepsies can have different patterns, and it's something that we can evaluate and that we are doing with the GRIN genes. The whole protocol that we've prepared, and I want to show it to you because I want to drive you through my results by showing you how we are doing it, because a lot of graphs sometimes are kind of weird to follow. I don't want you to look at the times or anything. The thing is that we are just playing with light. And in dark light conditions inside the recording chamber, they behave in a very stereotypical way. In the dark, they move more, and once the lights are on, they move less, and they always have this pattern. In here, we can evaluate their movement, their distance recorded, and also if they have any visual problem, and their visual impairment will also be detected in this, and will allow us to maybe perform more experiments to see what's happening really in this visual impairment. Afterwards, what we perform is an habituation. As if you remember yesterday with the Drosophila, with the flies, we had a flash of light and they moved a lot, and they habituated this movement. It's quite similar with this, but in here, it's not a flash. It's like we are tapping the plate. It's like if someone was hitting on the walls of this room. We will be really scared at the beginning, but then we will habituate, and we can assess this kind of behavior in the fish. And finally, as I mentioned, the convulsion pattern, by flashing the fish with light, we can see if they are moving, having this aberrant moves, and we measure it by the velocity, the speed where they are moving after the flash, and these kind of turns, this kind of movement aberrant that they have after the flashes. And now it's just, this is unpublished data, it's just a sneak peek of what we have so far. Focus on GRIN 1A, or GRIN 1A and B, because it's the obligatory subunit, and we started with that. When we observe their movement, they have a less movement. The mutant animals, when compared with the wild type, means that they don't have the mutation. They are unmutated, and we have less movement, so it will seems like they have hypotonia, as we can see in some of the patients. When observing the convulsion pattern, in the maximum velocity, we don't see many differences, but when we see this movement, to this kind of turns that they do, we can see that in the mutant, they seem to be turning more. And maybe it's not epilepsy, but it's some kind of sensitivity to light or to external stimuli. When going to the other GRIN gene, the GRIN 1B, we again see this hypotonia, this hypolocomotion, and when going to the epilepsies, in this case, it's quite clear. They have maximum speed higher, and these turns are higher in a very significant way. And when we combine these two genes, as I mentioned, and we have our pathology model, and in here, please focus only on the dark blue, because it will be the super mutant animals. We can see, even if it's slight, this hypomotility again. It's slight, but we can see these these epilepsies or convulsion patterns in the animals. But what was really interesting is that when applying this habituation pattern, the mutants had really a deficit in the habituation and more excitability to this external stimuli. And the idea is to perform, and we are doing it, but I don't have time, and also it's a lot of data, so I will not show everything in here, but we are doing this with all the mutants that we have generated so far. And the idea is to evaluate several different things in these mutants: locomotion, epilepsy, visual impairment, autism disorders, or autistic-like behaviors, memory and learning deficits, sleep disorders, that is something that we are already doing also, with the idea of perform these integration studies to take everything into account, not only one symptom or one phenotype, and to really characterize these models as a disease model. And the final idea is the identification of potential therapeutic targets, meaning that maybe not only with the NMDA receptor, but any other signaling pathways that are involved in these different symptoms that we are seeing, trying to find something to treat them. And my last part is something that I really like, and it's quite weird, I know, but we have set it up, the evaluation of gastrointestinal function. Since we know constipation and not chewing or not eating properly, it's a common symptom in the patients, and since the zebrafish are transparent and we can work with them, it made sense to see how their gut was working. So we performed this, we set it up, this experiment again. We breed the fish as we do with the behavioral experiments. We collect them, and at 7 days this time, because it's when the gut is more functional and its absorption properties are fully functional, we count the larvae, we treat them or not, and we feed them with fluorescent food. This fluorescent food can be seen in their guts, and we image and record their guts first time point. And after 16 hours, we record and image the same animals to see how this fluorescence has been or not reduced. And for you to see it, this is how the gut of a 7-day zebrafish will look under the microscope. You can see the blood circulating, and clearly they are super alive. And we can evaluate the peristaltic movements, how it's moving, the amplitude and the frequency of this movement, and compare it with the results related with the fluorescent food. These are the same animals. Here it's with bright field, and here it's with fluorescence. And we can evaluate how much have they eaten at the beginning, and how much have they be eaten in the end, how they are evacuating, they are getting removed of this feed after 16 hours. First experiments with pharmacological models of loss of function in NMDA receptors showed us that it seems that something's happening here that was quite interesting. And when performing the first experiments that are quite preliminary, but I wanted to show them to you, starting with GRIN1, we do 2 evaluations, how much they have eaten at the beginning and their digestive performance, meaning how much food is left there. And here we have the wild type, the animals without the mutation with the constipation control, our animals without the mutation without any treatment, and our animals with the mutation without any treatment again. In the GRIN1A, we can see that they seem to eat normally, but in the digestive performance, the lower is the number, the more constipated they are. And we can see that it's just preliminary, it's just one experiment, but the mutants seem to have constipation problems. And when going to the GRIN1B, they don't seem to have constipation problems, maybe a slightly decrease, but it's nothing significant. But when looking at how much they have eaten at the beginning, they have clearly an intake defect. Now we are working with the double mutants to see what's happening, but okay, this is something that we can see or in some of the patients, so it seems interesting. And if we find any treatment for this, it will be interesting to see what's happening with this kind of phenotype or symptom. And going to another model, that is the GRIN2A. In GRIN2A, we don't observe nor initial intake defects or constipation, but going to GRIN2AB, we observe both of them, that they have this intake deficit, quite significant, and also that they have constipation after 16 hours. All this data that, as I mentioned, it's quite preliminary and unpublished yet. Hopefully next year it's going to be everything published. It's to our final end that it's to find some molecules, some treatments for our patients. And with all this characterization, that it's a wide characterization, our idea is to test all the compounds that we can to find if something hits somewhere. Doesn't matter, it doesn't have to be everywhere, but maybe only on locomotion or only on the gastrointestinal function or whatever. And if it works, then probably move it to mice, to any mice model. And if it works and it finally works, probably moving to clinical trials. And I always like to finish with this meme, because I think it defines both our researchers and families. We're always saying, why is this happening? Why? And after research, after time, after sharing everything, yeah, that's why. Then more questions are unanswered, I know, but this is how we're always working. And everything we do is thanks to you, the families, for these events, for the funding. In my case, la Asociación Española de GRINopathies gave me funding to keep working for a few months before I started my PhD, and I kept working with GRINopathies, and I'm doing my whole PhD in GRINopathies because I'm in love with you. So and it's really possible thanks to you and these events and the sharing between researchers and between everything. And also to thank the groups, Clinics group, as I mentioned, we are a big company. I'm here. And without them, even if they are not working with GRIN-related disorders, we are working as a big team, and also with Xavi, and here, well, Xavi and Javi Terrientarich, my other director, Mireia, David Soto, and all the collaborators. Thank you for everything, and that's it.